In 1932, a group of scientists at Bayer were on the brink of something revolutionary. At the helm of affairs was Gerhard Domark, whose team had just synthesized Prontosil, the first commercially available sulfonamide antibiotic. This was groundbreaking at the time, as it became the first synthetic antibiotic that was potent yet relatively safe. It even won Domark a Nobel Prize. Bayer thought the patent on the drug would be their gold mine, and it was, but for a short while. In an unexpected twist of events, scientists discovered that Prontosil was effective in treating bacterial infections in living organisms but showed no antibacterial effect within test tubes. This puzzled researchers until the pharmacist Ernest Fourneau and his team at the Pasteur Institute in France uncovered the truth. Prontosil wasn't the real hero. It was actually a prodrug that the body metabolized into sulphonamide, which possessed antibacterial activity both within the body and in test tubes. This revelation was disastrous for Bayer, as sulphonamide had already been synthesized by the Austrian chemist Paul Gelmo as far back as 1908. With his patent long expired, the floodgates opened for every pharmaceutical company and private researcher eager to tweak its structure to create even more potent antibiotics they could patent for themselves. One such researcher was a certain Mr. Ruiz. He was searching for new antibiotics for typhoid fever when he stumbled upon an unexpected side effect. Some of his test animals developed low blood sugar. He speculated that sulfur drugs might lead to insulin secretion, but the medical community ignored his observation for a while. Soon, World War II ravaged through Europe like wildfire. As the war raged on, food shortages increasingly became more severe. People in cities like Montpellier resorted to eating whatever they could find, with some historians claiming that many survived on rotten food. As you can easily guess, the unsanitary conditions led to a deadly outbreak of typhoid fever. Thankfully though, sulfonamide antibiotics discovered almost a decade ago were becoming increasingly easier to synthesize and had become widely available. So doctors prescribed sulfonamide antibiotics by the thousands. The physician Marcel Jambaud of the Montpellier Medical School administered a new sulfonamide, an isopropyl thiodiazole, which had the code name 2254RP, to 30 typhoid patients. What followed was alarming. Several of the patients, already emaciated from typhoid fever, experienced a drastic drop in their blood sugar levels. This led patients into seizures, some slipped into comas, while a few sadly died. Jambaud was baffled. This wasn't a normal reaction to antibiotics. Determined to understand this anomaly, he informed top physiologists at the medical school where he worked, hoping someone could make sense of this finding. When Auguste Laubertiers, a physiologist at the Montpellier Medical School, heard the news, he immediately recognized that the symptoms Jambaud described were similar to those caused by excessive insulin. Decades earlier, in 1889, the researchers Oskar Minkowski and Joseph von Mering had demonstrated that removing the pancreas from dogs induced diabetes. 2 years later, Emmanuel Hedon at the Montpellier Medical School, in an attempt to further their research, performed a partial pancreatectomy where he left a well-vascularized fragment of the pancreatic gland and noticed the pancreatic remnants prevented the formation of diabetes in most of the animals. And in the few that still came down with diabetes, it was notably less severe. Subsequent research by Jean de Meyer postulated the existence of a sugar-regulating substance within the pancreas called insulin, a hormone Frederick Banting and Charles Best successfully isolated in 1921. Laubertiers at this time had extensively experimented on the long-acting insulin called insulin protamine zinc, also known as Prozinc or IPZ, on diabetic dogs. He had observed that high doses of IPZ induced severe and prolonged hypoglycemia, sometimes leading to convulsive attacks and comas. The similarities between IPZ's effect and the reactions seen in Jambu's patients were too striking for him to ignore. So he hypothesized that 2254-RP was somehow stimulating the pancreas to flood the body with insulin. To test his theory, Laubertias began animal experiments using the established Hedon's model in June 1942. He administered the thiodiazole compound to normal fasting dogs and observed a marked and long-lasting drop in blood sugar levels. However, when he gave the drug to dogs whose pancreas had been completely removed, there was no effect. In drugs with only a partially removed pancreas, the drug once again triggered hypoglycemia. The pattern was clear. The drug's effect depended on the presence of pancreatic tissue, which confirms that it stimulated insulin secretion. But Laubertias was not satisfied with his own genius. He wanted absolute proof. So he designed an insane cross-circulation experiment. In this procedure, he connected the pancreaticoduodenal veins of a normal dog to the jugular vein of a dog that has been made diabetic by alloxan, a chemical that destroys insulin-producing beta cells, which leads to insulin deficiency and hyperglycemia. To do this, he first injected dog A with alloxan. This destroyed its pancreatic beta cells and made it diabetic. Then, he made a connection between a major vein in the pancreas of another dog B and connected it to the jugular vein of the diabetic dog A. Now, the jugular vein is a vein that carries blood from the brain and head area to the heart. So now he connected a vein from the pancreas of dog B to the jugular vein of the diabetic dog A. This ensured that the blood gotten from dog B would immediately reach the heart of the diabetic dog A, where it can be pumped throughout the entire body before enzymatic degradation of insulin is allowed to occur, as endogenous insulin normally has a half-life of around 4 minutes. Then, he injected 2254-RP into dog B and noticed a reduction in the blood glucose levels of dog A. This could only have occurred if 2254-RP injected in dog B stimulated the release of insulin from dog B that transversed through the connected veins into diabetic dog A and caused a reduction in the sugar levels in the diabetic dog A. Elated, Laubertias documented his findings in a thesis written in French, where he stated that 2254-RP is an essentially insulin-secreting body. A French scientist had once again made a monumental contribution to diabetes research. He proposed that hypoglycemic sulfonamide could be used to treat forms of diabetes which were a consequence of the laziness of insulin secretory mechanisms. Sadly, Laubertias' work largely occurred during World War II, when German troops occupied France. His research then fell into the hands of German pharmaceutical companies who were reluctant to share their knowledge and kept the findings locked away. After Germany's defeat in 1945 and the partition of Germany into East and West Germany, the drugs were trapped in East Germany, where scientific progress and further research was delayed by political division. It wasn't until 1952 that a smuggled sample of Sulphonylureas reached West Germany, which reignited interest in sulphonamide antidiabetics. Pharmaceutical giants then scrambled to refine and commercialize the drug. Eli Lilly rolled out a urea-based sulphonamide called carbutamide, which became the first oral hypoglycemic agent. But Upjohn, who collaborated with Hearst, the makers of tolbutamide, ultimately won when Eli Lilly's version was found to be toxic. Tolbutamide was approved by the FDA in 1956 and was marketed as a treatment for diabetics who still had some pancreatic function, just as Sulphonylureas had intended. It was a game changer, but like all breakthroughs, it was imperfect. By 1970, long-term studies suggested that tolbutamide could increase the risk of cardiovascular issues, leading to its decline in popularity. However, scientists continued refining sulphonylureas, which led to the development of the second generation sulphonylureas in use today. Gliclazide, sold under the brand name Diamicron, was the pioneer's second generation drug. It was first synthesized by Berege at Servier Laboratories in France. It was aimed at reducing the cardiovascular risks associated with sulphonylureas. Berege achieved this by adding a bicyclo ring to the sulphonylurea parent chain. The antioxidant effect of this ring helps protect the vascular tissues from damage and tremendously reduce the cardiovascular risk observed in first-generation drugs. Soon, other second-generation drugs emerged, starting with glipizide, developed by Pfizer under the brand name Glucotrol. Glibenclamide, sold under the brand name Dalnil, was developed from a collaboration of two companies that are now part of Roche and Sanofi, while glimepiride entered the market under the brand name Amaryl in a quest by Sanofi to improve on the flaws of glibenclamide. Not much is known about the story behind their innovations, as these drugs were produced by Big Pharma with less serendipity associated with them, but more conscious structural activity modifications. These modifications, however, are not foolproof, as all the second generation sulphonylureas still come with a label that highlights the risks of cardiovascular issues in some patients. All sulphonylureas also carry the risk of causing severe hypoglycemia with overdosing, as is expected from excessive secretion of insulin. However, these risks are less likely to occur with the much safer glimepiride. Today, sulphonylureas can be used alone or in combination with other diabetic therapies, such as the combination of metformin and glibenclamide to form glucovans. Some modern diabetes guidelines have fallen out of favor with sulphonylureas, as their insulin secretion mechanism leads to an increase in the body's metabolism and a concomitant increase in the amount of carbohydrates absorbed, which then could get stored in the body as fat and consequently lead to weight gain. Many have argued that this effect in the long term adversely affect the prognosis of diabetes, as medication therapy tends to fare better in people when they experience weight loss due to exercise and diet changes. Regardless, we can all appreciate how a simple antibiotic experiment led to a medical revolution. The journey from Prontosil to Sulphonylureas is a testament to the unpredictable nature of scientific discovery. How one accident could pave the way for life-changing treatment. In our next installment, we'll explore how scientists modified various natural hormones to arrive at some of the modern diabetes treatments that help millions today.